Hi everyone, and welcome to our video on genetic control of body plan development. This is part of our Module 6 Part 1 series about cellular control, and what we'll be looking at in this video is specification references 6.1.1c, the genetic control of the development of body plans in different organisms, and 6.1.1d, the importance of mitosis and apoptosis as mechanisms controlling the development of body form. The first thing then is we need to understand a little bit about these things called homeotic genes. Now, when we're talking about the homeotic genes, these are the ones that are involved in the control of the anatomical development of an organism. So basically what that means is that they're going to be making sure that all of the structures that makes up the body of this organism will be in the correct location according to its body plan. Now, several of these contain what we call a homeobox sequence, and we can refer to those as homeobox genes. The homeobox sequence then is a 180 base pair sequence that's involved in regulating patterns of anatomical development in animals, plants, and fungi. Please remember those three: animals, plants, and fungi is the homeobox. Now, our homeobox sequence is going to code for something we refer to as the homeobox domain, and this is all about how these animals, plants, and fungi are actually going to develop. So that homeodomain sequence can actually fold up into a particular shape, and when it does that, it can bond to DNA, and as a result of that bonding, can regulate the transcription of the adjacent genes. That means it's basically acting as a transcription factor. So quite simply, that homeodomain sequence can then bind onto a particular region of our DNA and therefore control the transcription of other genes next to it. Before we go any further about this, there are two terms we need to learn carefully, because if you use these in the wrong context, you can cost yourself marks on the exam papers. So, we've been talking about homeobox genes, and they are found in animals, plants, and fungi. But we can also refer to some as being HOX genes. If we're talking about a HOX gene, this is only in animals. So if we're talking about animals, you could use homeobox or HOX. If we're talking about plants or fungi, you have to say homeobox genes. In terms of these homeobox genes, then one of their key features is that they are highly conserved. So that just tells us that across all of these organisms from a wide range of evolutionary distances, these homeobox genes are actually really similar. So basically these are not undergoing much change, even though we're talking about potentially very large periods of time and therefore a lot of evolutionary steps that could have taken place. So let's focus in on how these HOX genes actually control body plan development in animals. So the HOX gene itself is able to regulate that development of the embryo along what we refer to as the anterior-posterior axis, i.e. it's going to control the development along from head to tail of our organism. So what we're actually seeing here is something called collinearity, which is a sequential and temporal development of the body parts. And that's a really fancy way of saying they control what body parts grow where and when they grow. So basically what we're seeing here is not just a free-for-all, they don't just happen whenever they feel like it. It's all a very controlled process of which bits of the body are going to develop and when they develop. There's a really clear progression sequence in the development of the body. So the HOX genes are going to code for these homeodomain proteins, and as we've already mentioned, these are acting as transcription factors in the nucleus for activating other genes. And these other genes that they're activating are all to do with either promoting mitosis, apoptosis, or cell migration. So mitosis is obviously that division of the nucleus that leads to a proliferation of our cells. Apoptosis is our programmed cell death, and cell migration, relocating cells to key regions. And the Hox genes are actually regulated by messenger RNA from the egg's cytoplasm. When it comes to this process of mitosis, then what we have is the ability to obviously create new cells. Now, normal body cells are actually able to divide a limited number of times before they die. And the number of times they can divide before dying is what we term the Hayflick limit. And this is around 50 divisions. So what we need to be able to do is actually control this part of the cell cycle where we've got that mitotic division of cells occurring. And this is all regulated with those homeobox genes. The second aspect that we need to regulate is the process of apoptosis, which is basically programmed cell death. Now, you might think, why on earth do we want to kill certain cells? And the reality is that we've got a few scenarios where we actually need to break certain cells down during the development of the body. Now, let's delve into the process of apoptosis first of all, before we have a look at some examples of where this would actually be utilised. So in apoptosis, the first thing that we're going to see happening is the cytoskeleton needs to be broken down by enzymes. Because remember, the cytoskeleton is all about providing that shape to the cell. The cytoplasm becomes very dense with these tightly packed organelles as we're obviously breaking down more of the actual cytoskeleton there. And therefore, we're then going to be able to form these little things called blebs. Now, a bleb is a small protrusion from the cell surface membrane. And what we then see is the changes in our nucleus. So the chromatin is going to condense, the nuclear envelope is going to break down, and DNA is then going to be broken up into small fragments. The cell itself is then going to break into these vesicles, and those little vesicles containing the bits of the cell that remain are going to be ingested by the body's phagocytic cells. In terms of when we're going to see this apoptosis process taking place, then we will obviously have to have it controlled because we don't want it happening all the time. It would be pretty detrimental to an organism to just have random cells dying here, there and everywhere without any kind of structure or coordination. So we have cell signalling taking place. We can use cytokines from the immune system in order to obviously remove cells that have potentially been infected. We can use our growth factors, we can use hormones. What we're looking at here are chemicals that are going to be used in the organism to signal between one cell and another that these ones here actually need to be broken down. And the key example of where we can see this taking place in terms of the development of humans is when your limbs were developing. Going back to when you were a teeny tiny speck, then obviously you went through a range of mitotic divisions, grew into larger numbers of cells, then obviously those cells started differentiating, and we had these Hox genes actually controlling, working from your head going down to your tail, which bits were going to develop where and when, i.e. your arms were on where your arms should be, your legs are where your legs should be, etc. But in those early days, your fingers and your toes were not the individual little digits that you probably have now. Instead, they were actually very webbed together. So they were actually more joined, almost like little fins and flippers more than anything else. And what we then have is, as you go through that development cycle, you get to a point where we want them to separate. And therefore, all of those bits of webbing between your fingers and toes, they're just cells. We need them to die. So we need apoptosis to take place, kills those cells off, removes them from the area, and then you get individual fingers and toes. And obviously this isn't a perfect scenario in all humans, as we can see from the pictures at the bottom, where we do still have someone with two toes very much joined there. And if we look at the fingers on this, we can see that those two middle fingers there are very much still conjoined. Generally speaking, what we should find in our organisms is that the rate of apoptosis should equal the rate of mitosis. This obviously assumes that the organism is not growing, etc. If that's true, then clearly the number of cells that are dying is equal to the number of new cells being created, and therefore the number of cells in the organism remains pretty constant. If, however, we lose control of this apoptosis one way or another, then we're going to see some issues potentially occurring. If we don't have enough apoptosis, so less cells are dying than are being created, this could lead to the formation of a tumour. If, however, too much apoptosis is occurring, this is where we can see that loss of cells, so degeneration. And these are obviously certain conditions that we see in medicine where those loss of specific cells then leads to obviously medical symptoms. So hopefully apoptosis is happening at the same rate as mitosis for you, and therefore you're not having any tumours forming nor any degeneration. It's the happy sort of middle that we're aiming for. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over into the website where you can find a range of other resources to help you in your A-level biology studies.